Good day guys, welcome grade 11s. Today we're looking at life sciences and we're going to be studying invertebrates. What are invertebrates? I know these questions are going through your mind, but let's look at what we've done already. So you guys have probably we've done a whole lot of microorganisms, we've done a bit of plant diversity. Today we're going to spend some time looking at invertebrates and exactly what they are. So we're going to try and recollect some of that. We're going to go through some of the salient features of invertebrates, and then we're going to engage with some questions. And I hope you guys are going to engage with your teachers and get some interaction going. So let's get straight into it, guys. So what are invertebrates? And that's a key question today. So if we have a very quick overlook at what we're looking at today. Here we're looking at the classification of animals, and from your recollection of grade 11, grade 10 and grade 11 stuff that you've done already, we know that when we look at animals, we can broadly classify them into vertebrates and invertebrates. And what do we mean by this? Well, essentially we're looking at animals that have a vertebral column and a skull, and we compare them to those that don't have one. And essentially we refer to them as organisms that have a vertebral column, which is a backbone, and the other referred to as invertebrates. And these are essentially organisms that do not have a backbone. So essentially we're focusing today on the organisms on this side. We refer to them as the invertebrates. So cool. So what are we looking at today? So we're looking at the salient features of all of these and why these organisms are characterized as protozoa, annelids, and so on. So if we broadly look at this classification system, you'd see that we've got a host of different organisms, and when we compare them, there's an increasing degree of complexity. So what we're going to look at today is the characteristics that are used to identify the organization or the body plan of these organisms and how they are classified. And as we look at these, we will see that the organisms are becoming more complex in their body plan or becoming more specialized. And essentially we're going to look at these over the next couple of weeks and look at how invertebrates are classified, their characteristics, and how they are of economic use or benefit to us. So let's get straight into the lesson today. So what are the key concepts that you need to look at? So when we look at invertebrates, what do we mean? We refer to animals that have a distinct backbone. And by that I mean a spinal column or lacking a spinal column. So these are animals that lack a spinal column. And we can classify organisms based on the presence of their backbone and some of the other features that are often used to classify them accordingly. So we're going to spend some time looking at the presence of these features and how these are used when identifying and classifying some of the invertebrates. So what are these features that we're looking at? Essentially today we're looking at 5 features, and those 5 features we will track across the phylum, and we will look at how they increase in complexity among different animals. And essentially what we're talking about today are the body of symmetry, the type of gut, the type of body cavity, segmentation, and cephalization. So with these, we're going to spend some time looking at essentially what each of these features are in terms of how to identify them in the organisms and how to use these to determine the degree of complexity and the degree of relatedness with other organisms having similar characteristics. So what we're looking at today is these key features and how they are used to distinguish organisms. When we look at these organisms, we can look at them according to the different phyla in which they are. So if we refer to the term phylum, phylum refers to a large group of classification of organisms under which they show a specific group of characteristics which we can classify them into. So what we're interested today is the body plan, and essentially the body plan refers to the way in which the body is structured, and that's based on these key features which we are going to unpack in a little while. So what do we mean by the key concepts of body symmetry? So when we look at body symmetry, essentially we're saying, well, every organism has either a pattern of symmetry or a manner in which we can compare the left and right hand sides. So what does body of symmetry refer to? So it refers to the possibility of dividing an animal in one or more directions and getting 2 identical halves. So let's rephrase that. It is looking at the possibility in which we divide organisms in one or more directions to get 2 identical halves. And by the way, is it possible to be able to divide every organism to get two possible halves? Well, we're not too sure, but let's look at some examples. So based on the ability of being able to divide organisms, we have three main categories of identifying symmetry in organisms. We have, if you look at these diagrams, we have an organism here that shows bilateral symmetry, and by that you can see a line or segment that goes down the organism. And this essentially shows that the organism can be cut into two equal halves that mirror each other. So when we compare the left and the right halves, we can see that they are identical. Here we have a polyp, and this polyp has got several lines of symmetry, and these lines of symmetry cut the organism in a manner that creates mirror images. And notice that being a spherical object or being a cylindrical object, it has got multiple lines through which the organism can be cut into equal halves. And such an organism is referred to as an organism showing radial symmetry. And essentially, guys, we're looking at the radius of the organism. So when we look at this organism, we're looking at the radius in which the organism can be cut along. So often you'll see this as a characteristic of organisms that are cylindrical or that are rod-shaped. Okay, cool. And here we've got a strange organism here which appears to not have any symmetry no matter which line that we decide to cut it along. So I'm going to try and cut this along different planes, and you'll notice that no matter which plane I decide to cut the organism, it is not going to generate any mirror image. So these organisms are referred to as asymmetrical or showing no symmetry. And it is a key feature that we would see with some of the simpler organisms that are not as diverse as some of the complex organisms. So what I'm going to do now is, I do have some specimens with me, but let's look at these, understand these concepts thoroughly first before we look at these examples. So here we're looking at, we discussed what bilateral symmetry is. We said that these are organisms or animals that are divided down the middle of the body in one place to produce two identical halves, and the word two can be linked to bi, and hence we refer to organisms as having a bilateral symmetry. We also looked at the second example was an organism that is round and that could be divided in many ways and generally around the radius to produce identical halves along the radius, and we refer to these as radially symmetrical. So look for the radius and look at whether the radius generates lines of symmetry, and that would be a key feature of an organism that is radially symmetrical. And the last example was an organism that, when cut in no matter any direction or plane, produces no two identical halves. So what are we saying? So I'm going to grab some organisms here. Here we're looking at, I've got a starfish, belongs to the group Echinodermata, and if you look at this, guys, although it appears to be symmetrical, if we plan to cut this along the radius, does it produce sides that are mirror images of themselves? And if you decide to cut this along this axis, you'll notice that it does produce the left and right replica images. So here's an organism that, depending on the plane in which we cut it, it can produce radial symmetry. However, not all organisms do that. Here we're looking at a sea urchin, and here's a classical example of an organism that can be cut along the radius to produce mirror images and multiple different lines of segmentation. Or to look at this organism, and you guys would probably have seen this in your National Geographic videos and movies. Here's a sponge, and it belongs to one of the first groups of organisms in the invertebrates. And if you look at it, no matter which direction we cut this in, it cannot produce identical halves or mirror images of each other. And we refer to this as an organism being asymmetrical, having no symmetry. So cool. So we've looked at symmetry. Let's look at the next concept. Organisms can also be classified according to their gut system or a type of gut that they have. So what do we mean by type of gut? So some animals have only one opening to the gut through which food is taken in and also eliminated. So we refer to those organisms, that passage of food, as the gut. Humans, if you look at, have two openings. We have the oral opening, and we have an anterior opening and a posterior opening, and that is the opening through which we ingest food and egest food or excrete food, and we refer to that as an organism having a through gut. However, guys, not every organism has a through through gut system, or in other words, they have a common opening. And if we look at the hydra here, guys, it has a common opening for the mouth through which it will ingest food as well as it will excrete or digest food. And refer to this organism as an organism having a blind gut. And by that I mean it has a single opening through which it uses to both food to enter as well as for waste to leave. So that's a classical example of an organism having a blind gut. And if you were to think about this, this would be a system that is not effective because it's using a common opening to both ingest food and to eliminate food. So again, not a very evolved characteristic, something that is very simple that would be found in some of the simpler invertebrates. However, if you look at the more complex form of a gut, you'll find that it has a definite anterior opening which is referred to as the mouth, and we have a posterior opening which is referred to as the anus. And through this passage we find that food moves through one end and is released to the other end. And this is a classical example of an organism having a through gut system where there are two openings. Essentially what we're saying is that this is a very evolved system, a more complex system, because the organism can feed and release waste at the same time. Cool. We also need to look at the type of body cavity, and to that we refer to the coelom. So the coelom is the cavity inside an organism. A coelom basically is lined with cells from the mesoderm. It is important because it keeps the internal organs separated from the outer layer of the body so that the internal organs can move and grow on their own. So we have a distinct coelom or coelomic cavity that is lined by a mesoderm. Okay. Animals that have a coelom are called coelomates, and animals that do not have a coelom are called acoelomates. So we're going to look at these as a feature of organisms. So having a true body cavity is referred to as having a coelom, and those that do not have one are referred to as acoelomates. However, there are some organisms that do not have, and refer to them, or do not have a true coelom. We would refer to them as pseudo-coelomates, and we will look at some of these examples as we get through to some questions. Also, we can also refer to the layers that are present in animals, in the bodies of these organisms. So there are two layers that are present in an organism, and we refer to those two layers as organisms being diploblastic, having two layers. If there are three layers or more, and generally there are three layers maximum, these organisms are referred to as being triploblastic. So we are triploblastic, we have three layers. In an embryonic development, we find that there's an endoderm, mesoderm, and ectoderm. We're going to look at these in these examples below. So when we look at examples of organisms having different coeloms or body cavities, we have an acoelomate. So an acoelomate would be an organism that does not have a true body cavity. So it has a body covering from an ectoderm. It lacks a defined coelom, but we also have a digestive layer formed from the endoderm. So this is an example of flatworms being acoelomic. We also can look at some of your coelomates, and coelomates are organisms that have a true body cavity, and here we're seeing a distinct coelomic cavity here that the organism has, and that is covered by surrounded by layers filled with a mesoderm and an endoderm, as well as an external outermost ectoderm. So we can see a distinct layer, and those layers are the ectoderm, the mesoderm, and the endoderm. And finally, we're looking at organisms that do not have a true coelom or coelomic cavity, and we refer to them as your pseudo meaning false, and as I said, coelom, a coelomic cavity. And here if we look at these organisms, they do have a cavity, but due to the cavity being lined by endoderm and lacking an outer a mesoderm on the inside, we refer to them as being a pseudo-coelomate. Cool. Let's move on to one more feature before we go into a break. Segmentation. Often we find that organisms have body parts that are segmented. Here we're looking at how the body is divided into different segments, and we refer to that term as segmentation. And if we look at these examples here, guys, we'll find that there are some organisms that clearly show distinct segmentation, whereas others that do not show. So S- body Segmented animals consist of a series of body units which differ from other animals. In earthworms, and most segments look alike from the outside, and we clearly see that they look similar. However, a spider will have two different body parts, which is each composed of specialized segments. Each of these three parts of the insect body have a head, a thorax, and an abdomen. Here we're seeing a segmented body composed of different parts with specialized functions. Here we're looking at, again, images of organisms that are flatworms. They have a flat body, non-segmented. Here we're clearly seeing a millipede showing a segmented body with multiple segments, each having specializations of different parts. And here we have roundworms that show a round body with no distinct segmentation to their body. So that's based on the concept of body parts being segmented. And finally, the last key feature we're looking at is cephalization. What is cephalization? It is basically the concentration of organs, especially the sensory organs, around an anterior end of the animal. And these are generally a concentration of nervous tissue around the head, around the opening of the mouth. And we see this among organisms that are generally bilaterally symmetrical. So cephalization refers to the development of a brain or an area of specialization which has a high concentration of nerves which control and coordinate nervous functioning and the body function. So cephalization, again, we said, is the concentration of sense organs and nerve cells around the anterior opening of the organisms. Here we're looking at a starfish, and here you clearly see a nerve ring with some kind of nerve that run along the radius, and we refer to them as radial nerves. Here we're looking at other flatworms and leech and insects that have a brain, a very primitive brain, a very simple brain. However, it is still found at the anterior end of the organism, as well as we see that in the leech, and we see that in most of our insects where there is a distinct brain present at the anterior end. So guys, we've looked at the salient features, the 5 main features. We're going to have a short break, and then we're going to look at some questions. Welcome guys, hope you had a good break. What we're doing now is we're looking at some questions, and I'm going to go through these questions and give you guys some time to have a look at these questions. And I think it's important that you work through them, and then we can go through these answers. And it's an essential part of you being able to consolidate your understanding. So let's get straight into these questions. And most importantly, we're going to look at terminology, and that's a fundamental concept, a way of understanding the concepts in this topic. So let's look at question number 1. So I'm going to read through these questions so that you can have a heads up for them and work through them. Right, so give the correct biological term for each of the following descriptions. So question 1.1 refers to animals that can be cut into mirror images in one plane only. Question number 1.2, animals which can be cut in any vertical plane through the central axis to give you mirror images. Question 1.3, animals which are irregularly shaped. Question 1.4, the presence of a definite upper and lower surface in animals. Question 1.5, animals that remain attached to the substrate for most of their lives, and the key word is attached. So guys, this should take you about 2 minutes. So what I'm going to do is I'm going to give you a 2-minute time to work through these questions, and then when you get back, we're going to go through some of these answers. So guys, have you worked on these? I'm sure you had some fun. Hope you had a discussion with your friends. Cool. So let's look at some of these answers, and I hope you've got answers. And if you haven't, guys, so I'm here to help you. So we look at these questions again, but I'll look at each one of them and go through the answers with you. Right. And as I said, it's very important, and as you would have seen before we went on this, I looked at the questions, I underlined the keywords, and that will help me to answer these questions. So again, 1.1, animals that can be cut into mirror images in one plane only. So essentially what we're saying is, if you look at an organism that can be cut in one plane only, what is that? And we discussed this as our first example, and we said that these are organisms that are bilaterally symmetrical. So we refer to them as being bilaterally symmetrical. So again, organisms that have one plane in which they can be cut into two equal images or mirror images, we refer to them as bilaterally symmetrical. Hope you guys got that. So that was quite a good one to start off with. 1.2, animals which can be cut in any vertical plane through the central axis to give you mirror images. And the key word here is mirror, and it's look at the plural images. So how often would you get mirror images? It has to be an organism that can be cut along the radius into several segments to produce mirror images along the radius. And guys, if you remember, the word radius should help you, and hence we refer to these organisms as having radial, or they're radially symmetrical, okay, or radial symmetry, symmetrical. Okay, so it's important that you explain the concept clearly. Cool, so hope you guys got that correct at least. All right, let's move on to the next one. I hope you guys are enjoying this. Animals which are irregularly shaped. So animals that are irregularly shaped, and we looked at this. So if we decide to cut this along any plane, guys, sorry, we're not going to be able to get lines of symmetry. So we refer to these as being asymmetrical. Okay, so again, we discussed this as a characteristic feature of the organisms that are less evolved. Some of the simpler porphyrians, they do not have any line of symmetry, and we refer to them as being asymmetrical. So guys, let's look at the next question. The presence of a definite upper and lower surface in animals. So what does this mean? Guys, if you look at any organism, and I'm going to draw a classical example of a leaf, which is often easy for us to relate to, guys. When we look at a leaf, it has the upper surface. Now, that upper surface is referred to as the dorsal surface. Okay, however, it also has the ventral surface, or the surface below. So the dorsal is the upper surface, and the lower surface is the ventral surface. So what is the term that describes organisms that basically have a definite upper surface and a lower surface? So we refer to these organisms as being dorsally, so dorsoventrally. So I'm just going to erase that for you quickly. So we refer to these as being dorsoventrally flattened. So these have both an upper end and a dorsal surface. So again, the key words are dorsal end and the ventral surface. Okay, cool. Let's move on to question number 5. So hope you guys have got 4 out of 5 so far. On the last one, let's see. Animals that remained attached to a substrate for most of their lives. And guys, you would have seen an image of the sea anemones and that are attached. You guys would have watched Nemo, and here you find an organism that is attached to a definite substratum and does not move for most of its life. It's classically found of clams, your shellfish. These are attached on a substrate, and they basically rely, and even your polyps and corals, they rely on the current to bring in food. And we refer to these organisms as being sessile. And sessile is the term that refers to organisms that are attached and remain stationary. So these were the 5 questions. I hope you guys have got these all correct. But let's look at some more questions. And these are more terminology. But what I think is, I'm going to work through these with you, and I'll explain these concepts as we work through them. So let's move on to these. So give the correct biological term for each of the following descriptions. Right, let's see. And you guys are going to help me and help the friend sitting next to you. Let's underline keywords. The accumulation of sense organs at one end often leads to the development of a head. And guys, we discussed this. We said that, think about it, when organisms start showing the development of sense organs at one end, and it leads to the development of the head. Right, so remember organisms like they have a cephalothorax. So what does that mean? A distinct head and a body. So we refer to this process as cephalization. Again, key concept when we're looking at salient features. So the development of a distinct head or an area that can respond by nerves to the rest of the body, or control center. We refer to that as cephalization. Well done to those of you that got it. Let's look at 1.7. Animals that arise from cells growing, cells arranged in two germ layers. Guys, two germ layers, so we have to refer to this as being diploid. So we refer to them as being diploblastic. So organisms that have two layers are diploblastic. Again, so we refer, and those layers we're referring to are the mesoderm and the ectoderm. And we're going to look at these in a bit detail through the next session. Okay. Animals in which all tissue develop from three germ layers. Guys, three germ layers, triploblastic. So we're referring to three germ layers. So these would show the mesoderm, the endoderm, and the ectoderm, and the ecto. And if you want to remember them correctly, so meso is the middle layer, endo is the inner layer, and ecto would be the outer layer. And refer to them as being triploblastic organisms. Cool. And let's see the last question, now the second last question. A kind of skeleton that results from muscles working against fluid-filled body cavity. And guys, remember we're dealing with invertebrates, and these invertebrates do not have a skeletal system, so they rely on structures that give them body shape or that allow them to bring about movement. And if we look at most of these invertebrates, especially your aquatic invertebrates or some of the terrestrial, they often rely on water filling into body tubes or their cavities that give them shape. So the kind of skeleton that where body tubes are filled with water is referred to as the hydrostatic skeleton. So we refer to that as the hydrostatic skeleton. Remember that we have the endo and the ectoskeletons, which are skeletal systems that have an internal skeletal system, or we have those that have on the outside referred to as the ectoskeleton. Let's look at the term that describes organisms with two digestive openings, and that takes us to an example would be the earthworm, where you see a distinct anterior opening in the mouth and a posterior opening in the anus, and refer to that as organisms having a through gut system. And that's essentially referring to a pathway of food traveling through from the mouth to the anus. So cool. So we've looked at some good terminology questions. Let's get straight into some of the more intricate questions that require a bit of differentiation and integration. So I'm going to take a few of these questions and possibly, give you some time to look at these before we go into a break. Okay, so I'm going to read these for you. Differentiate between 2.1, a true coelom and a pseudocoelom. 2.2, a blind gut and a through gut. 2.3, radially symmetry and bilateral symmetry. So guys, 3 questions for you guys to look at. I think I can give you about 3 minutes to look at these. Work through them, try and write your answers down, and we'll get back to these solutions after 3 minutes. So guys, I'm sure you had fun working those out, and obviously it's just relating to whatever we've just done. So I know that you guys are busy working, you've enjoyed this session. I think you guys deserve a break, get some water, and we'll get back. So we'll take a short break, and then I'll see you guys at the end of this break. Welcome back, guys. So I know you've enjoyed your break. Let's look at some of these solutions. I know you've got some interesting answers, but let's look at what are the salient features of these answers. So again, let's look at what a true coelom is. So a true coelom refers to a true body cavity. So it has a true body cavity, and that body cavity basically has, it's lined. And what is it lined with? It's basically lined with a mesoderm and an outer ectoderm plus an outer ectoderm. So that is a key feature of a true coelom. So it has a mesoderm and an outer ectoderm. So that's characteristic of organisms that have a true coelom. So that's a true coelom. What is a pseudocoelom? Remember guys, the word pseudo, and often people say, well, you know, he has a pseudo name referring to a false name. So the word pseudo basically refers to false. So this would be an organism having a false body cavity, and that cavity is basically lined with only an outer, it's got basically only mesoderm, okay, and it lacks an outer ectoderm. Cool. Let's move on to the next question. A blind gut and a through gut. Again, repetition of a concept. So guys, blind gut would refer to an organism that has a single opening through which it will basically ingest, so a single opening through which it ingest and excrete waste products. Okay, so a single common opening through which it will ingest. And if we look at a through gut, guys, a through gut is basically referring to a gut in which there is an opening at the anterior end called the mouth, and there is a posterior opening at the end called the anus. So that is at the end. So you have a distinct opening at the anterior end, and you have an opening at the posterior end. We refer to them as having an anus and the mouth. So it's a through gut. Cool. So I hope you guys got that one correct. Let's look at radial symmetry and bilaterally symmetrical. So let's go with bilaterally symmetrical. So bi would refer to an organism that can be cut along a plane, and that produces to give you two identical halves. So we're looking at two identical halves or mirror images. Okay, and radial would refer to organisms that can be cut along many planes, and that produces several or many identical halves. Okay, and that is we will see in organisms that often have radial symmetry. Cool. So that was quite an easy question, but it required you to bring in and compare concepts. Let's look at some more questions, and these are based on looking at the content of the question and identifying what's right and wrong. So guys, so we're looking at true and false questions, often questions that you can easily score marks in. So each of the following statements could be true or false, so they could be true or false. In the space, write true or false, and if false, you're going to write the wrong word followed by the correct word. So that's important. You've got to identify the wrong word and you've got to put in the correct word. So let's look at these very quickly. So phylogeny is the study of how groups of organisms evolved. Now I know that evolution is the study of how groups of organisms have evolved. So the incorrect word there is evolve, so that's definitely going to be false. and the incorrect word there is evolved, and the correct word should be classified. So I'm just going to write the correct word as being classified. So we're saying that the incorrect word is evolved and the correct word should be classified. So that's quite easy. Let's look at the next one. Birds and mammals are ectothermic chordates with endoskeleton. Now guys, you would look at the development of organisms based on their body and their salient features, but also we need to refer to some of them in terms of their body temperature. And those that have a constant body temperature are referred to as endoderms, and those that have a temperature that fluctuates based on the environmental conditions, we refer to them as being ectothermic. So previously we would refer to them as being cold-blooded, now we refer to them as being warm-blooded. Are birds and mammals cold-blooded? No guys, if we feel our body temperature, we know that it is constant, it remains around 37.5 degrees Celsius. So that is a warm-blooded animal. We refer to us as being endotherm. So the incorrect word here is ectothermic, so that's a false statement, and the correct word should be endothermic. Okay, cool. Let's see the next question. 3.3. The cylindrical polyp is the free-swimming body form of the phylum Cnidaria. Guys, Cnidarians are not free-swimming, so they are basically that's the incorrect word, so that statement's false, and we should be saying that they are the sessile forms of the phylum Cnidaria. Okay, 3.4. In triploblastic organisms, the layers are the ectoderm, mesoglea, and endoderm. Guys, we haven't mentioned the word mesoglea. It's the ectoderm, endoderm, and the mesoderm. So obviously that's an incorrect statement, and the correct word should be the mesoderm. Cool. I'm sure there must be a true somewhere here. Let's look at the next one. Earthworms increase the nitrogen, phosphorus, and potassium content of soils. And guys, if you'd seen with lots of rain around the country, lots of the earthworms would be enjoying this, and that's essentially what they do. Increase the nitrogen content, phosphates, and potassium content. So that's a true statement. So well done. So we've worked through this. Let's look at some more questions. So this is quite an interesting question. Basically, I've put in a little image here to help you understand different sections that are present. So let's look at the question. Label the following diagram using different terms describing symmetry. Also identify the ventral and dorsal surface sides, the posterior and the anterior ends using the letters A to D. So guys, so what I did was I put in this image here just to basically help you. So here we're looking at a squirrel, and that's cut along, you know, it's a three-dimensional diagram that shows you possibly different angles to this. Here we're looking at a squirrel, and if you were asked to cut a section along the sagittal plane or transverse plane, that's essentially referring to this angle that we're going to cut through. If we're looking at a sagittal plane, this angle will create bilateral symmetry. So if we cut the organism this way, we're going to generate a left half and a right half that is bilaterally symmetrical. However, if we cut along the transverse plane, we produce sections that are not symmetrical. Okay, when we look at an organism from this end, we refer to that as the frontal view. From the end, that's the posterior end. So it's important that you're able to orientate. Where will dorsal be? So we're looking at the upper surface, and we look at the ventral surface. So these are key features that you should be able to use when identifying. Here we have an organism, and guys, if we were to cut this up, so what section of this is it? So we're looking at basically a section that shows the left and right. So this would be cutting it along that's sagittal plane, and we're getting an organism that shows bilateral symmetry. So that's bilateral symmetry there. And here we have the posterior end, so that A is going to be the anterior end, and here we have the posterior end there, so that's going to be the posterior end. And we can look at, so the ventral surface would be the side that has the vertebral column, and we have the dorsal surface, which is there. Cool. This is the exact question that the diagram that the question relates to. So here we're having an organism being cut, and it's being cut along the radius, and we refer to this as having being radially symmetrical. And so here you clearly see it being cut along the radius, and each of these segments produces identical section. Guys, if we look at this organism, here we have a common opening, so that's going to be the anterior end, and this would be basically the posterior end of this. So cool. So we've looked at an organism that you're able to identify different sections of. So guys, we're going to look at the last question. I'm going to spend some time going through this, but essentially it's a comparison of some of the structures that we've already looked at. It's important that you look at the mark allocation for these questions. Here we've got to differentiate again between terms. So we're looking at diploblastic and triploblastic, and guys, you'd seen that diploblastic are organisms that have 2 layers, triploblastic are organisms that develop from 3 germinal layers, so the mesoderm, the endoderm, and the ectoderm. I want to look at this question before we break off. So it's a very nice question to actually end the session off with. Explain the advantages of cephalization and why is it seen in bilateral animals. So guys, if we look at a bilateral animal and if we compare that, if we just draw in a stick figure of a human, okay, not very good at stick figures, but here we see the development of a central nervous system. So we see the CNS. Now why is this a significant adaptation for organisms that are bilaterally symmetrical? So in this question we've got to explain that cephalization basically is the centralization of a nervous system around the anterior end of the organism. And you guys know that having a head, you're able to firstly think, coordinate, and having this system called the brain or the central nervous system that brings about coordination. That's essential in terms of an organism surviving. And being bilaterally symmetrical, you know that when we stand up, we can coordinate activities based on the way we walk, we bring about coordination. But more than that is that having it at the anterior end means that you're able to respond to situations, you're able to see. So if we isolate ourselves from the examples of invertebrates, but we look at all the other invertebrates and look at how they have got an anterior end which shows cephalization. So if we look at the earthworm, if we look at most of them, so having an anterior end means that they're able to respond to changes in their environment. And that's a key feature of living organisms, being able to respond, being able to feed. So they have a distinct anterior end that allows them to pick up stimulus. Being bilaterally symmetrical means that that central nervous system allows them to be able to respond and bring about coordinated activities. And hence we see this as a distinct adaptation of organisms being bilaterally symmetrical. So key features of an organism being bilaterally symmetrical is that they're able to respond to changes, they're more alert, they are able to bring about coordination between the left and right hand sides, and that is called nervous coordination, which we see, and that is why we study the nervous system as we see progression. So we've looked at, guys, in this session, we've looked at invertebrates, we've looked at the key features of symmetry, of having a coelomic cavity, of looking at segmentation. And I think these are important salient features for us to understand and remember when we look at how we classify your invertebrates. So I hope you guys have enjoyed this session. Go through these, work through some of these questions, build up a definition, a list of terminology, define these concepts, write them down and refer to them when you're preparing for an assessment. It's important that you have these with you and you constantly refer to them. So from my side, I hope you guys have enjoyed the session. Look at these questions again, spend some time revising. All the best.